Good afternoon. So this is the final lecture of the cancer series. Today we're looking at cancer genetics and causative agents of cancers. So to recap, what is cancer? Cancer is a disease of cells. You can get it in skin cells, nerve cells, gut cells, red blood cells. And new cells are made via cell division. And cell division is required for growth, healing, and replacing of old cells. So in the past minute, your body has made at least 300 million new red blood cells, 12,000 million new gut cells, and 40,000 new skin. So let's have a look at the loss of normal growth control. So when you have normal cell division, the cells grow, but when the cell is damaged, you have no repair, and this can result in cell suicide or apoptosis. The loss of normal growth control from the cancer cell division, this eventually happens from the first mutation, second mutation, third mutation, fourth mutation, and uncontrolled growth. So that's basically what happens in loss of normal growth control. There's another video that I've done previously in the loss of cell control cycle, which leads to cancer. The types of cancers in the body, so some common carcinomas are lung, breast cancer, then there's sarcomas, fat, bone, muscle, then there's leukemias, blood cancers, and lymphomas, lymph nodes. More than 200 types of cancers are present. So let's have a look at the cancer prefixes. So you have adeno, meaning a gland, chondro, which means the cartilage, erythro, which means red blood cells, hemangio, blood vessels, hepato, liver, lipo, fat, lympho, lymphocyte, melano, pigment cell, myelo, bone marrow, myo, muscle, osteo, bone. So looking at some terminology of the cancers, leukemias are cancers of the blood or bone marrow. Sarcomas are cancers of the connective or supportive tissue such as bone, cartilage, fat, muscle, blood vessels, and soft tissue. These are quite rare. Carcinomas are cancers that arise from epithelial cells, which include breast, liver, lung, stomach, etc. So carcinomas develop from epithelial cells. So specifically, a carcinoma is a cancer that begins in the tissue that lines the inner or outer surfaces of the body, and that generally arises from cells originating in the endodermal, ectodermal germ layer during embryogenesis. Carcinomas can affect the breast, lung, prostate, and colon, and are among the most common types of cancers in adults. They are rare among children before adolescence. Children are more affected by cancers that are not carcinomas, such as leukemias, lymphomas, and sarcomas. So looking at the classification of carcinomas in more detail, adrenocortical carcinoma affects the adrenal glands. Thyroid carcinoma affects the thyroid gland. Nasopharyngeal carcinoma affects the nose and the pharynx. Malignant melanoma describes skin cancer. Skin carcinoma is anything other than melanoma, and other carcinomas including those affecting the salivary gland, colon, appendix, lung, bronchus, cervix, urinary bladder. In comparison, sarcomas arise from transformed cells of mesenchymal origin. Therefore, malignant tumours are made from bone, cartilage, fat, muscle, vascular, hematopoietic tissues, and are by definition considered sarcomas. To put this in a bit more detail, mesenchymal cells are multipotent stem cells which can differentiate into a wide variety of cell types. So all tumours which are benign and malignant have two basic components. They are proliferating neoplastic cells which constitute their parenchyma, and supportive stroma made up of connective tissue and blood vessels. Parenchymal cells are the proliferating leading edge of neoplasms, but the growth and evolution of neoplasms are critically dependent on their stroma. An adequate stromal blood supply is required, and the stromal connective tissue provides the framework for the parenchyma. So looking at the molecular basis of cancer, so the body is always copying DNA and making new cells. You can see here, to recap, the base pairs, adenine, thymine, cytosine, guanine. You've got the DNA, double helix, etc. So what actually causes cancer cells to become cancerous? So look at the molecular basis. So you have the non-lethal genetic damage which lies at the heart of carcinogenesis. And this is genetic damage or mutations acquired by the action of environmental agents such as chemicals, radiation, or viruses, or maybe it is inherited in the germline. So it can also be caused by viruses or bacteria, chemicals, radiation, or cell processes. So cancer is often perceived as a disease that starts for no apparent reason. While scientists don't yet know all the reasons, many of the causes of cancer have already been identified. Besides intrinsic factors such as heredity, diet, hormones, scientific studies point to key extrinsic factors which have a role in a cancer's development. Chemicals such as smoking, radiation, and viruses or bacteria, diet. These factors cause DNA damage and/or cellular and or cellular macromolecules. So, look at the mutations here. You've got the normal genes compared to the syndrome base changes, additions, deletions, syndrome base used to be a substitution. You've got a translocation as well, where one part goes to another part of the chromosome, and the duplications. And development of cancer is usually a multi-stage process. Let's have a look at colorectal cancer as an example. So this has separate mutational events. So it is normal, goes to adenoma, then adenocarcinoma, then metastasis. So you can see here the genes that are involved, etc., in this and the time that it takes. The chemicals that cause cancer include carcinogens, such as cigarette smoking. So cigarette smoke is, I'm actually not a big fan of smokers, to be honest. I don't really like smoking, I think it's disgusting. But you have 4,000 greater than 4,000 chemical substances, and greater than 60 are known to be carcinogenic, which include arsenic, benzene, cadmium, nickel, and 2-naphthylamine. 80% of lung cancer deaths caused by smoking. 1 in 3 of every cancer's death is caused by smoking. So you also have cancer-causing agents in the workplace, such as asbestos, benzene, lead or dust, naphthylamine, vinyl chloride, and wood dust. So you can see the occupations that are present in this here, and the type of cancer that it causes. In the event of asbestos, lung cancer; benzene, leukemia; lead or dust, nasal bladder cancer; naphthylamine, bladder cancer; vinyl chloride, liver; and wood dust, nasal cancer. They are infectious agents such as viruses and bacteria. Some of the viral genetic information carried in nucleic acids and inserted chromosomes of infected cells, and this causes cells to become malignant. So here's an example of bacteria in stomach cancer. The bacterium Helicobacter pylori causes stomach ulcers, has been associated with development of cancer. So people infected with pylori are at increased risk for stomach cancer. So you can see a list of viruses on the left hand side in the top left, or the top right. Epstein-Barr virus, human papillovirus, hepatitis B virus, a human T-cell lymphocytic virus, and the type of cancer that they cause, liver cancer, etc. Can affect many types of cancers. So inherited conditions that increase the risk for cancer include xeroderma pigmentosum, which results in skin cancer; Wilms tumor, kidney cancer; familial adenomatous polyposis, colon and rectum cancer; and BRCA1/2, breast or ovarian cancer. So what are the targets of DNA damage? There are 3 classes of normal regulatory genes which are the principal target of genetic damage. That includes the growth-promoting proto-oncogenes, the growth-inhibitory tumor suppressor genes, and the genes that regulate programmed cell death or apoptosis or DNA repair. So this probably requires collaboration between several oncogenes and several tumor suppressor genes. Important to note that no single genetic change is common to all cancers. So proto-oncogenes are natural genes that regulate cell growth with the potential to become oncogenes. Genes for growth factors, C6 encodes the PDGF gene. The gene for receptors, EGF2, which results in ERB2. Genes for signaling intermediates and cell cycle transducers such as cyclins. And oncogenes, so genes that code for mutant forms of signaling molecules that regulate proliferation, differentiation, and survival. Genes coding for the following can mutate to cause cancer: growth factors, growth factor receptors, cytosolic transducers, nuclear transducers. So mechanisms of dysregulation of proto-oncogenes in cancer. So proto-oncogene, if you look here, you have 2 types. You have the deletion in the mutation encoding sequence that was involved in hyperactive protein made in normal amounts, such as RAS. Then you have the gene amplification, such as normal protein is greatly overproduced in MYC. Here's the oncogenes operating cancer. You can see here the hyperactive RAS protein with the product of oncogenes. Tissues signals on its own. Looking at tumor suppressor genes, so mutated tumor suppressor genes results in loss of growth control. So copies of tumor suppressor gene on both homologs are mutated. So here's an example of another tumor suppressor gene that having an effect, retinoblastoma. You see here in the diagrams how retinoblastoma operates. So DNA repair genes, there are genes that ensure each strand of genetic information is accurately copied during cell division or cell cycle, Mutations in DNA repair genes lead to an increase in the frequency of mutations in other genes such as proto-oncogenes and tumor suppressor genes. For example, breast cancer susceptibility genes BRCA1, BRCA2, hereditary non-polyposis cancer susceptibility genes MSH1, MLH1, PMS1, PMS2 have DNA repair functions. Their mutations result in tumorigenesis. So cancer tends to involve multiple mutations. So benign tumor cells grow only locally and cannot spread by invasion or metastasis. Malignant cells invade neighbouring tissues, enter blood vessels and metastasize to different sites. So here the mutation inactivates suppressor gene, cells proliferate, mutations inactivate DNA repair genes, proto-oncogenes mutate to oncogenes, and more mutations, more genetic instability and metastatic disease.